Hi everyone. My name is Kalina Belemezova. I work in the genetics lab in the Dr. Sterev Hospital. I'm also an assistant professor in the Medical University of Sofia. Today with my colleague from Vidacom, Slavena Tudorova, we are going to address the issue of HPV testing, but through the eyes of the people who actually work in the lab and do the testing. So to make this a little bit more interesting in the beginning, I would like to start with a quiz. So I'm gonna ask you to scan the QR code with your phone. We prepared a few questions for you. Okay, did everyone did it? Okay, let's start. So I'm gonna read just the question. You can read the answers yourself. How is HPV typically transmitted? Easy one. Okay, 5 people voted, 8. Someone's making a joke, I think. Okay. Did everyone vote? Okay, let's see next one. Okay, so 100% through sexual contact, including vaginal, anal, and oral sex. Okay. Next one. Do you know if there are different strains or types of HPV, and if so, how many are there? Let's see here what we have. Okay. Almost everyone thinks there are more than 200. Okay, we have slight different answers here. Did everyone vote? Okay, you'll find the answer in the presentation and you can tell me.
More than 50, more than 200 means more than 50 and more than 100. So
yeah, around, let's say around 50, right. Around 100 and more than 200. Okay, let's put it this way. Okay. What are some common symptoms of HPV infection? Again, easy one. Okay, then if we are done with this one, I'm gonna start the other one. Which types of HPV are considered high risk for causing cervical cancer? Okay. Oh, sorry. Okay, so 100% said types 16 and 18. True or false? HPV can also cause cancers in areas other than the cervix? Absolutely, yes. Okay. Okay. Then, can HPV infection be cured with medication? Okay, we have different answers here. We can discuss this later after the presentations and after the workshop. Okay. And last one, not to bore you. Okay, which of the below can influence and on, there's a type mistake here, sorry, on the HPV DNA test result? Yeah, we're going to discuss the sample collection, sample storage, and how important it is to actually get the right results and be sure that this is the true result. Okay? Next one. Does HPV viral load has any clinical significance? Okay. someone, okay, maybe someone said no. Most people said yes. We'll find out soon. Okay. What does the SIC number mean? You'll see it in the results we have prepared for you. Maybe you've heard this, but if not, you'll have to guess. (50-50) Okay. Okay. Are you waiting for someone else to vote? Okay. Okay, then we're done with this part, and I'm gonna tell you a little bit more about the HPV virus. Its way of its life cycle, its way of escaping the immune system, because I'm an immunologist, and immunology is my first love. And then my colleague is gonna tell you about the actual test, the sampling, and how to interpret how to read the results. We have prepared here some results straight from the PCR machine, and we would like to give them to you and think about them, and while we are presenting, and maybe afterwards we can discuss them and you can tell us what you think about these. I promise to be short and not to bore you too much with biology details. So, you all know this, but again, it is estimated that approximately 80% of the sexually active individuals will contract the papillomavirus at some point at their lives by the age of 50. HPV 16 and 18 account for about 70% of cancers of the cervix, vagina, and anus, and for about 30 to 40% of cancers of the vulva, penis, and oropharynx. And although HPV is an important cause of cervical cancer, it's not a sufficient one. There are multiple cofactors that are required in order to have the actual HPV transition to cancer. Currently, more than 200 types of HPV are known that belong to the family of Papillomaviridae. You probably know this. Just quickly, the HPV can infect the basal epithelial cells of the skin or the inner lining tissues, and it is categorized as cutaneous types or mucosal types. And according to its oncogenic potential, we categorize them as high-risk viruses, which include 13 types. Of these, 16 and 18 are the most famous one and the most common one. The low-risk viruses include 6, 11, 42, 43, and 44, and these are the ones that are responsible for the genital warts. Infection with the most oncogenic types 16 and 18 increases the risk of developing cervical cancer by more than 200-fold, and every year around 500,000 new cases of this type of cancer are diagnosed in the world, over 90% of which is a result of persistent HPV infection, and at least half of which are caused by HPV 16. Like I said, there are other cofactors that are necessary in order to have this oncogenic potential actually realized, and these include other infectious agents like chlamydia or herpes simplex virus type 2, vitamin deficiencies, and of course immunodeficiencies. The virus is relatively small, it's non-enveloped and contains a double-stranded, closely circulated code DNA genome. It's about 8,000 base pairs, which is not important for you, but what's important that it has 8 or 9 open reading frames that are named early or late according to the protein they encode. It's protected by a capsid formed by 2 proteins, L1 and L2, which are crucial for the entry of the virus in the host cell and which are important for the design of the vaccines. The early genes that we call E1 to E7 have multiple roles in the viral genome replication, cell cycle entry, immune modulation, and virus release. Their expression occurs throughout the viral life cycle, but reduces during the later stages of the infection. This is why they're called early. And in contrast, the late genes that are for the capsid proteins L1 and L2 are highly expressed during the later stages of the infection. The assembly of the virus occurs in the cell, in the nucleus of the cell, and what's important to know is that the proteins E6 and E7 are the ones that are associated with the integration of the viral DNA into the host genome and malignant transformation and progression to cancer. These proteins contribute to the cancer initiation and progression by altering the cell cycle, and the methylation, as we already heard, the telomere maintenance, as we also heard from the previous lectures, and induce DNA damage and genomic instability. They interact with the tumor suppressor genes, the p53 and the retinoblastoma. About the life cycle, shortly, the HPV, as any other virus, has its own strategies to escape from the immune system, one of which is that it infects in the basal epithelial layer, the cells that are in the basal layer, and that happens when these are exposed through some microlesions or some other sort of lesion. The HPV genome replicates in the nucleus of the basal cell layer, and there the reproduction is considered non-productive. The virus establishes itself as a low copy number, and it can stay there and reproduce there. Active cell division makes the cell move towards the top layer and undergo differentiation. In the differentiated keratinocytes, it switches to another type of replication. We call this rolling circle mode of DNA replication, and it allows it to copy very quickly and to actually make the virion and the viral particles and release them. Like I said, the E6 and E7 proteins are the ones that are connected to the oncogenic potential because they interact with p53 and retinoblastoma, which are very important for the cell cycle regulation. And once the cell cycle regulation is disrupted, the cell can start passing the cell cycle control points easily, and the cell cycle shortens. The immune response to HPV is actually the same as to any other type of virus infection. But, like I said, because the virus is in the basal layers, the immune response is not very well established according, the humoral immune response cannot be very well established because the virus is in the basal layer. Generally, we have IgG and IgA antibodies that are the most abundant one. The antigen-presenting cells, of course, in the layer are there, and they're going to detect the virus and are going to process it and present it to the adaptive immune system and release cytokines and local inflammatory factors that are signaling that there is an infection actually there. Like I said, the HPV is, as any other type of virus, has its own strategies to escape from the immune system. One of which is already, I told you about the base layer. The other thing is that it blocks the interferon expression and secretion, which is very important. The interferon response is very important for the antiviral immunity in our body. So this is one thing. And the thing that most viruses do, they suppress the antigen processing and presentation to the surface by MHC class 1. So they are hiding from the cytotoxic T lymphocytes this way. Just a little bit about the nature of the vaccines. You know this, you know there are vaccines, but how they actually work. 3 prophylactic HPV vaccines have been approved that target the high-risk HPV types. These are based on recombinant DNA technology. We have purified L1 protein, the one from the capsid, and that's assembled into an empty shell. So we have basically an empty capsid without the DNA inside. And this capsid elicits an immune response, and we have neutralizing antibodies. It's important to have more than 1 dose of vaccination because this guarantees that we'll have a polyclonal immune response, which means we'll have different antibodies, neutralizing antibodies that are specific for different epitopes of this L1 protein. This is the reason why we need to have more than 1 dose in order to mimic this polyclonal immune response. We also have a therapeutic HPV vaccination, and these are designed in a way that we don't need actually neutralizing antibodies. We need to work on the cell-mediated immunity through the cytotoxic T lymphocytes. This is about the HPV and the nature of the HPV. Now my colleague is going to tell you about the testing, actually. So don't forget that good research starts with happy cells.
Hello everyone, my name is Slovena and I'm an application specialist at Read.com.